Sequence of events for synaptic transmission. First, in the presynaptic neuron, axon potens reaches the terminal site, thereby opens the voltage-gated calcium channels, and calcium is influx from the synaptic cleft to the cytoplasm of the terminal neuron. This calcium helps in the docking of the vesicle to the nerve terminal membrane and releases the neurotransmitter. This is magnified view showing the detail of neurotransmitter release. Calcium ion triggers synaptotagmin and snare protein to induce membrane fusion and neurotransmitter release. Now neurotransmitter release and diffuses into synaptic cleft, which later binds with the postsynaptic receptor in the postsynaptic neuron, and this will generate two types of potential: local synaptic potential, that can be either excitatory postsynaptic potential EPSP or inhibitory postsynaptic potential, that is IPSP. Later, neurotransmitter is removed from the synaptic cleft and this will terminate the synaptic transmission. Binding of neurotransmitter to postsynaptic receptors. Neurotransmitter rapidly and reversibly bind to receptors on the plasma membrane of postsynaptic neuron. The activated receptors themselves may be ion channel, which designates them as ionotropic receptors. Alternately, the receptor can be also metabotropic type, in which receptor may be indirectly influence ion channels through G protein coupled receptor or second messenger system. In either case, the result of binding of neurotransmitter to the receptors is the opening or closing of specific ligand-gated ion channel to the postsynaptic plasma membrane of a neuron, which eventually leads to the change in membrane potential in that postsynaptic neuron. Now let's talk about the two types of potential changes in the postsynaptic neuron, EPSP or IPSP. EPSP, that is excitatory postsynaptic potentials. If the potential change in the postsynaptic membrane during synaptic transmission is toward depolarizing, then the potential is known as EPSP, while IPSP, if a potential of a postsynaptic membrane is carried towards hyperpolarization, then the potential is called IPSP. EPSP is because of the two excitatory neurotransmitters. There are others as well. The main is acetylcholine and glutamate. So this excitatory neurotransmitter will generate EPSP by the influx of sodium or calcium ions in the postsynaptic neuron. While other way, the inhibitory neurotransmitter, for example GABA and glycine, it has two mechanisms. It will either opens the chloride channel and leading to the chloride influx, or it will open potassium channels, thereby leading to the potassium efflux. In both cases, there is a decrease in membrane potential more than that of the resting membrane potential, and it will later inhibit the postsynaptic neuron. While in the EPSP, many EPSP they can summit, and finally this will excite the postsynaptic neuron.